Hi guys, welcome back to the channel. In this video, I will cover the DNA, genes and chromosomes topic of the A-level biology curriculum. So let's get started. So first, covering the eukaryotic DNA. So you need to know that the eukaryotic DNA is contained within the nucleus. So if we're looking at the eukaryotic cell, the nucleus is inside that cell, and inside that nucleus is where the DNA, the eukaryotic DNA, will be contained. And the eukaryotic DNA is linear. So what this means is when we're looking at the DNA, it will have an end to it. So this will be an end and this will be the other end. So for example, the prokaryotic DNA, which is circular, there is no end to it. So this is what makes the eukaryotic DNA linear. And the eukaryotic DNA is very long, and it is associated with some proteins called histones. Looking at these are the histones. So what they do is they help the DNA to coil together, so it becomes really compact, so more information can be stored in less space. So this is the job of histones. And the eukaryotic DNA forms chromosomes. So as these chromatins, as this DNA keeps coiling, and what happens at the end is they end up forming this structure which is called a chromosome, and this is super compact. And the eukaryotic DNA contains introns within the gene. So these are non-coding regions, and these introns are within the gene, so they're inside the gene. And the non-coding multiple repeats, which are sometimes also called mini satellites, now they are between genes, so they're basically outside genes. So looking at a picture, so for example, if we have this gene, the mini satellite would be between the genes. So if that's a gene and that's another gene, so the mini satellites are between those genes. So they are not inside the gene, they're outside the genes. And if we're looking at the introns, so this is our gene, and the introns, so these non-coding regions, so these gray ones, will be within the genes. So we have these exons, which are the coding regions. They are the only coding regions, but we have introns within the genes, which are non-coding, and we have the non-coding multiple repeats, which are between genes, and they are also non-coding. Alright, so now looking at the prokaryotic DNA. So the prokaryotic DNA is inside the cytoplasm. So like we had the eukaryotic DNA, which was inside the nucleus, now in prokaryotic DNA there is no nucleus, so the prokaryotic DNA is just free floating inside the cytoplasm. And the DNA is circular this time, so it's not linear, it's circular. So circular doesn't mean it has to be a perfect circle. What it means is that there is no end to that DNA. So like a circle carries on forever, you can't find an end to a circle. That bacterial DNA and that prokaryotic DNA is circular. It's not linear. It won't have an end to it. It has, because it's circular. So it's much shorter than the eukaryotic DNA. It's very short, and there are no associated proteins. For like the eukaryotic DNA, we had those histones. For prokaryotic DNA, there are no associated proteins. And these prokaryotic DNA do not form chromosomes. Again, one of the reasons is because they do not have those histones, so they are unable to form the chromosomes. And they have no non-coding DNA. So in eukaryotic DNA, we had those the introns and the mini satellites, but in prokaryotic, all the DNA contains exons, so there are no non-coding regions, and this is one of the reasons that the prokaryotic DNA is short, because it doesn't have all them the non-coding regions in this. And the prokaryotic DNA also contains these plasmids, which the eukaryotic DNA doesn't have, and these plasmids also have their own DNA. So the eukaryotic cells, mitochondria and chloroplasts. So you need to be aware that the mitochondria and the chloroplast in a eukaryotic cell, they're both short, they're both circular, and they both are not associated with proteins. So they're quite similar to what a prokaryotic cell would look like. So they're circular, they're short, and they do not have any histones. So this is a similarity between the eukaryotic cells and the prokaryotic cells. So now just looking at all the similarities and differences between the eukaryotic and the prokaryotic DNA. So the eukaryotic and prokaryotic both have an identical nucleotide structure. Remembering that nucleotide structure of DNA, we have that deoxyribose sugar and that phosphate group and that nitrogenous base. So that's the same with prokaryotic and eukaryotic DNA. They both have the same nucleotide structure, and the nucleotides are joined together by the same phosphodiester bond. So if we have another DNA coming along, the DNA will be connected by these phosphodiester bonds, and that's the same for both eukaryotic and prokaryotic DNA. And the DNA in mitochondria and chloroplast is similar to prokaryotic DNA. We have just looked at that. So now the differences between the prokaryotic and eukaryotic DNA. So prokaryotic has, again, that circular DNA, which has no end to it, but eukaryotic DNA has that linear DNA, which does have an end to it. So the prokaryotic DNA is much shorter than the eukaryotic DNA. One of the reasons being that it does not have any non-coding regions. The prokaryotic DNA is not associated with any proteins, whereas the eukaryotic DNA does have proteins which are called histones, which helps it to coil. And the prokaryotic DNA does not form chromosomes, whereas the eukaryotic DNA does form chromosomes. So again, the prokaryotic DNA has no non-coding regions, whereas the eukaryotic DNA has those introns and the mini satellites. So now looking at a gene is basically a base sequence of DNA. So this is the definition that you're required to know. And a gene can code for amino acid sequence of a polypeptide. So when the amino acids join together, they form a polypeptide, and a gene is what codes for these amino acids, and also code for a functional RNA, so such as the ribosomal or the transfer RNA. So gene can code for both the amino acid sequence as well as a functional RNA, and a gene occupies a fixed position on a particular DNA molecule, and this is called a locus. So for example, let's say we were looking at the gene for hair color. So if this was the gene for hair color, then this would be the exact position on both of these chromatids. So the hair color gene would not be there for one and there for one. It would have that fixed position, and that fixed position is called a locus. So a sequence of 3 DNA bases is called a triplet. So for example, looking at this gene, so we have this long gene, and if we are looking at a triplet, so this would be 3 DNA bases. So this would be a triplet, this would be another triplet, this would be a triplet, and this would also be a triplet. So this has 4 triplets. So every 3 DNA bases is called a triplet. And the triplet codes for a specific amino acids. So what happens is that the DNA is transcribed into mRNA, and then it's translated into these amino acids, these individual amino acids. And so what this is saying is that, so the triplets, so for example TAC, would essentially turn into a specific amino acid. So from the mRNA to the amino acid, but essentially the specific triplet code would code for a specific amino acid. So for example, TAC, it won't be able to code for, let's say, the WAL or Leo or SER. it can only code for that met. So the triplet codes code for a specific amino acid. And you need to know that the genetic code is degenerate. So some amino acids can be coded for by more than one triplet. So for example, looking at this example, so we can see, so if we're looking at phenylalanine, so there, so we can see that U, so U can code for phenylalanine, and whereas the UUC can also code for this. So there are essentially two codes coding for the same amino acids, and that's why the genetic code is degenerate, because the same amino acid, so phenylalanine in this case, can be coded for by more than one triplet. And the genetic code is universal, so in all organisms, in all the different species, the same codon codes for the same amino acid. So codon is basically the word we use for mRNA, and so when the transcription occurs, the triplets converted into a codon. We'll be looking at it in the next video. But essentially, the genetic code is universal because the same codon can code for the would code for the same amino acids in all the different organisms. The genetic code is also non-overlapping. So what this means is that each base is part of only one triplet. So for example, looking at our original gene, so for example, when we have that TAC, and that would code for an amino acid, and that TAC would only code for that one amino acid. So for example, if I were to say I have TAC coding for an amino acid, and then I have that CCA triplet coding for an amino acid, this can't happen because then these two would be overlapping. So what happens is that TAC would be only used for one amino acid, then the CAC would be used, then AAC would be used. So we can't have the triplets overlapping to code for an amino acid. You need to be aware of the fact that in eukaryotes, much of the DNA is non-coding. So in eukaryotes, on average, 99% of the DNA is non-coding, and only 1% of the DNA is actually coding for an amino acid. So we have the non-coding multiple repeats. So these again were called mini satellites. And these are between genes which do not code for anything. And we also have, so within genes we have the exons which do code for something. So these are the only things that do code for something. And the exons are separated by the non-coding introns. So again, we have the genes in there, the genes being the white ones, and between the genes we have those black bits which are the non-coding multiple repeats, or the mini satellites, and they do not code for anything. So these are between the genes. And then inside that gene, so this being the gene now, and we have these grey bits. They're not really grey, but just to show you that we have these introns which are non-coding regions, and they do not code for anything, and exons are the only bit that do code for amino acids. And much of the eukaryotic DNA, we would only have the non-coding region. In prokaryotic DNA, we don't have any non-coding regions, which is why it is very short. Thank you so much for watching this video. If you did like this video, don't forget to subscribe to see more of these, and you can watch my recent videos by clicking on the links popping up. Thank you.